So Nobel Prize has been awarded to them for the CRISPR Cas9 genome editing technology. So origin, from where it is all origin. So without before understanding that what is CRISPR, we have to see that from where it has originated. So this is your bacterial cell, and when a virus attack on it, so it releases its viral DNA inside the bacterial cell. So inside the bacterial DNA, there are two types of the sequences. First one is your CRISPR, which is cluster regulated interspaced short palindromic repeats. So it's palindromic because it's from the 5 prime to 3 prime if we read it, or if we read it from the 3 prime to 5 prime, it is all same. So this is your CRISPR cells, and this is your spacer, which we can call CRISPR locus. So bacteria, it will do, it will cut down the small fragment of the viral DNA and store in this CRISPR locus region, or we can say it spacer region. So this is your CRISPR array. Next, we can see what are the components of the Cas9, CRISPR Cas9. So here are the guide RNA. Firstly, the guide RNA is a RNA molecule that directs the Cas9 protein to a specific target. Okay. After that, it will guide Cas9 nuclease enzyme, and it will work as a molecular scissor and cut down the DNA fragment. Target DNA, which we have to cut down, and PAM sequences, it's the nucleotide sequence present adjacent to your target DNA. Okay. Without PAM sequence, Cas cannot bind or cleave the DNA. Okay. So now how this CRISPR-9 works. So firstly, the guide RNA, this is your guide RNA. So firstly, the guide RNA will come and it will attach to the DNA target sequence. Once it is attached, the Cas9 enzyme will come and it will cut down this target DNA target sequence and the PAM sequence, which is the adjacent to your target DNA. Once it is cut down, it has two options. First, we can add here desired DNA sequence or add for repairing. यहाँ पे हम लोग desired DNA sequence को add कर देंगे for the repairing purpose of the DNA. After that, its mechanism of action of repairing is of two types. One is non-homologous end joining, and the second one is homology directed repair. Directed repair. So non-homologous, in this ये directly join हो जाएंगे. The broken DNA and ends directly joins. In homology directed repair, cell repairs a double stranded DNA breaks. Okay, cell repairs double stranded DNA break. So this is the mechanism. Here no templates were used, but here templates were used from which this DNA sequences copy the your DNA base pairs, and precise genome editing happens. Okay. After that, here, what are the types of the CRISPR system? So here are the two types of the CRISPR technology used. One, the class 1 CRISPR system. Second one is the class 2 CRISPR system. So it is based on the effector proteins. Effector proteins is your, guide proteins. Guide proteins ke base pe hota hai. Okay. So your class 1 CRISPR system uses multiple Cas proteins. So here we use multiple Cas protein for the target recognition, and once the target is recognized, it is cleaved down. So its example, its types is type 1, type 3, and type 4. And class 2 CRISPR system, here we use only single large Cas protein for DNA and RNA cleavage, and in type 2 we use Cas9, type 5 we use Cas12, and type 6 we use Cas13. After that, we can see here some of the software and bioinformatic tools and its simple use, and whether they are free or paid. So firstly, it is Benchling, which is used to guide the RNA and CRISPR experiment planning, and it's a paid software for students and for the academics. Free for students and academic, it's free, but it is paid for industries, we can say. SnapGene DNA sequence visualize कर सकते हैं, we can cloning कर सकते हैं, and the genus prime sequence analysis and genome editing research. Okay, CLG Genomics Workbench, which is used for genome and CRISPR data analysis. CRISPR is free, which is used for the single guide RNA design and off-target prediction. Single guide RNA design and your off-target prediction. So next one is ChopChop. So ChopChop is used for CRISPR target finding and primer design. Okay. So next we will see the why CRISPR Cas9 is better than the traditional editing tools. So traditional editing tools, we use TALEN and ZFN. So TALEN is your transcription activator like effector nuclei, and ZFN use your zinc finger nucleus. So here we use zinc finger protein, and here we use TALEN transcriptor activator like effector nucleus protein. So here we have to design proteins for every single targets. But for the CRISPR, we can just program our guide RNA and we can get our desired DNA sequences. Okay, so this is why it is better and comparing its cost also, approximately design is very less, 50 to 500 US dollar, comparing to Talon and ZFN, which is called traditional editing technology and tools used for this. So this is why the CRISPR is used over Talon and ZFN. Okay, so now talking about the ground level reality, the FDA has approved one therapy named Cas9 therapy, which is given by the Excel company, and disease it targets sickle cell disease and beta thalassemia. And the technology are used the CRISPR Cas9 and first FDA approved CRISPR therapy, which is approved in 2023. Okay, so there are so many clinical trial drugs are there, and anti-LA in 2001, ATTR, amyloidosis, hereditary angioedema, genetic blindness, and leukemia, HIV infection, sickle cell disease. So here all we have used CRISPR. Okay, so talking about the applications. Okay, so application if we see in the gene therapy, in the sickle cell anemia, cystic fibrosis, Duchenne muscular dystrophy, and in cancer therapy, we can use here engineering immune cells and development of CAR T cells. Okay, CAR T cell therapy, and drug discovery and development, identification of new drug targets, understanding disease pathway, faster screening of potential drug molecules. After that, infectious disease management, detection and targeting of viral genome, like in HIV, human immunodeficiency virus. So if any virus came and it will immune system को अगर वो affect कर रहा है, तो हम कुछ ऐसे जेनेटिक मॉडिफिकेशन करेंगे जिससे वो HIV और कोविड 19 हेपेटाइटिस इन बी ऑफ टेक्निकल लिमिटेशंस में ये हो जाता है कि कभी कभी जो म्यूटेशन है वो ऑफ टारगेट होने लगता है, डिलीवरी चैलेंजेस आर देयर, इनकम्प्लीट एडिटिंग आर देयर, एंड पैम सीक्वेंस dependency is there. Okay, so ethical concern if we talk about, because we are playing with the we are going around the DNA genes, so there are always ethical concern for the CRISPR-Cas9. So firstly here the germline editing. So the long term effect on human may occur, and genetic changes may occur made in embryos can pass on the future generation. If we have made any genetic changes to the babies, that will pass to their future generations. So designer babies are there. So here are intelligence, appearance, strength will be possibly selecting for these. After that, safety and unintended mutation may occur. Only wealthy population could have the access of this kind of expensive therapies, and regulatory and legal challenges are always there. So the recent advances, if we talk about, we can use the base editing. Suppose here the C, we can convert it into the T after this therapy. Okay. So here the base editing, we can see there is C, the cytosine, and we have converted is the only single base we have changes after the T is tyrosine. Okay, after that prime editing, search and replace. We will search, हम यहाँ पे search करेंगे, उसको replace करेंगे. CRISPR diagnostic की if we talk about CRISPR diagnostic, so therapies like Sherlock and detector enables rapid disease detection. So Sherlock is I think used for SARS-CoV, COVID-2 and Zika virus, and detector enables rapid detection tools. They are फिर AI assisted CRISPR. Here we will assist our guide RNA with the AI so that it cannot cause off-target mutation. Okay, improved precisional editing could be there. So CRISPR in cancer therapy, CRISPR engineered immune cells are being developed for target cancer treatment. Cas12a2 system can selectively destroy cancer cells while sparing healthy cells. Okay, so here we can see it is invading only cancer cells, and it's not touching even the healthy cells. After the next generation CRISPR, we will use Cas. Apart from Cas9, there will be Cas12, Cas13, and TIGR, and clinical gene therapy. So I have already told you Cas9, which is already FDA approved, FDA approved therapy, and it is a FDA approved and very useful therapy. Okay. आफ्टर दैट हम देखेंगे इसके फ्यूचर पर्सपेक्टिव से पर्सनलाइज मेडिसिन एंड ट्रीटमेंट फॉर जेनेटिक डिसऑर्डर, एडवांस्ड कैंसर थेरेपी लाइक इन CAR-T cell and regenerative medicine, next generation gene editing. After that, your development of disease resistant crops. So with the help of this, we can use like a long term अगर प्लांट में कोई डिसीज है तो हम उसका जीन एडिटिंग करके उस डिसीज को रिमूव कर सकते हैं वैसे. सो दीज आर द रेफरेंसेस फ्रॉम वेयर आई हैव टेकन द डेटा, नेचर पब्लिकेशंस, एंड लेटेस्टली आई हैव टेकन artificial intelligence to advanced CRISPR based genome editing technologies, nature reviews. So thank you so much for your attentive listening. And one thing that Black Shelton has said, that one thing that you can't fake, it's chemistry. So I hope you guys like this video. अगर आपको ये वीडियो पसंद आई हो तो बिल्कुल इस वीडियो को कीजिए सब्सक्राइब और चैनल को कर लो लाइक. Thank you so much, guys.